Hi, everybody. Welcome to Improving Qualification, Validation, and Development with UHPC. My name is Kevin Wood. I am the Director of Consulting Services at Voltaiq. And with me today is Stephen Glazier from NOVONIX. To introduce myself, my name is Kevin Wood. I am the Director of Consulting Services at Voltaiq. Do a lot of things, really kind of helping try and get engineers to understand and get the most out of their battery data that's possible. This includes everything from helping with design of experiments and kind of doing things on the technical front to helping with education and doing educational seminars and providing resources to really help people understand. There's kind of a couple important parts of, understanding and working with battery data. One is that you have to have the test set up right. You have to understand and be able to perform the analysis. And then you have to really be able to understand all of that information and kind of, I hope, help our customers really make sure that they're able to do all three of those really, really well. So before joining Voltaiq, I was a CTO of a startup company, was a professor for a number of years, working in kind of next generation research. Excited to be here with you all today, and I'll pass it over to Stephen.
Hi, I'm Stephen Glazier, Director of Technology at NOVONIX Battery Technology Solutions. We're up in Nova Scotia, Canada. Really here, I focus on both our internal and external R&D projects with our partners and customers that utilize our cell testing equipment here, our cell line that we have to make pilot prototyping cells, really bring our own and our customers' technology to life, do proof of concept, work with them, and then also to focus on our ultra high precision coulometry roadmap. And that's really what I'll be talking a lot about today, is using ultra high precision test data to make the most out of your development, qualification, validation work streams. I've been with NOVONIX for about a little over 6 years now. Before that, I was at Dalhousie University here in Halifax, Nova Scotia, working with Dr. Jeff Don on my master's and PhD, where I covered a lot of different topics, including using a lot of high precision metrology to figure out what was going on in different degradation mechanisms and anode, cathode, electrolyte, et cetera. So that's me.
Awesome. Thanks, Stephen. All right. So we're just going to kind of go ahead and jump right into it. To kind of kick things off here, what does quality, validation, and development, how does that really affect you? So to kind of, start off, quality issues become orders of magnitude more expensive as you kind of progress through the battery life cycle, right? So when you're research and development, right, they may still be, somewhat costly, but those costs are much smaller than after you've gone towards product development and cell manufacturing. And then once you're starting to integrate those into a system, and then as you go to in-field, right, there's kind of this progression of exponential growth in terms of how costly product issues or development issues are. So if there's a product or a critical flaw in your cell, it's much better to find that out in your research and development phase than after you've gone to cell manufacturing, right? I mean, that just kind of makes sense. But, really what we're trying to get to is we want to try and identify these issues as early as possible, right? So to improve quality, right, you really kind of have to address two fundamental challenges, right? One challenge is that you've surfaced quality issues too late. So that's a key challenge, right? You need to surface those issues as soon as you possibly can. The second is really focus on helping understand why that quality issue occurred in the first place, right? Sometimes people could just say, oh, it was a random defect, but you really want to try and understand why that happened and how you could try and mitigate that risk as much as possible. And so really to improve quality, you kind of have to surface issues early and really understand the origin and, just have more knowledge as to why that quality issue occurred in the first place. So, to really try and, address this, right, and improve quality, you really need to get to root cause analysis sooner, okay? So that's a lot of what, Voltaiq helps with, that's what NOVONIX helps with, is really trying to identify anomalies in your battery data, right, or in the heartbeat of your battery, or however you want to think about that, right? We want to try and identify those anomalies as quickly as possible and alert you to those, and then give you tools to help you understand and perform root cause analysis, right? Trying to get towards what I would call mechanistic understanding. Like, what is the mechanism of why this anomaly occurred, and how can I try and prevent that anomaly from occurring, moving forward? So, the earlier we can do that, the better off we will be. And the more we're going to save your company money, the better your product will be, all of those things. Everything improves the earlier you can get towards mechanistic insight. So if you heard me talk before, right, you may have heard me talk about what I like to call the battery iceberg, right? And this is really, you could kind of think of the iceberg as there's the tip of the iceberg that stands out, kind of over the water. And that's a very small percentage, but it's a very visible percentage. But then kind of what leads to that iceberg sticking out of the water is everything that's underneath the water, right? And I kind of like to talk about what's above the water. That part of the iceberg is the performance. And then everything that's kind of underneath are all the processes and the things that led you to that performance, right? So things are really, really visible, like cycle life, capacity, safety, voltage fade. There's some of these big metrics that we like to talk about, right? It's like, oh, how many cycles did you get to 80% capacity? And that's a very visible metric, and it's very, very important, right? However, what leads to that is really the process that's underneath this, right? This is going to be the quality of your materials, your slurry formulations, those sorts of things, the SOPs that you're using, all of those things are more the process that's going to lead to that performance. But some of the things that people don't necessarily think about are, the data that you're getting. If you're only analyzing a very small percentage of the data that you're actually generating, right? If you're an R&D company or anything like that, you're just wasting lots and lots of money if you're not analyzing all of that data. So you want to have good access to data. You want to have good data pipelines. You want to have good process controls. You want to have metadata, being able to link information from one part to the other, right? You kind of want to have all of this, right? It's the processes and the processes on the manufacturing side and on the material side and on the data side and the analysis side. You kind of want all of those things to work really, really well so that you can, positively affect the performance and the things that are, very, very visible. So, the challenge here is that a lot of times we just focus on performance, and really process is a really, really key piece to actually making the performance better. And, if we just focus on the performance, we kind of miss the actual things that are going to help us get to that mechanistic insight and actually make our systems much more sustainable and much better. So what can we understand from our battery data, right? So now let's say that you've got good quality equipment, right? We're talking about NOVONIX and their ultra high precision coulometry equipment, right? That you have maybe good data pipelines. That's things that Voltaiq can help with, those sorts of things. But what can you actually get from battery data? And that's part of what we're going to be talking about today. Stephen's going to talk about UHPC, but I'll just give you kind of a warm up to some of the things that we can get from battery. I like to think about a battery when a battery cycles is it leaves behind a fingerprint. It leaves behind a unique identifier. Every battery data point that you get, or every charge profile that you get, is very, very slightly different. But those slight differences can give you lots and lots of clues as to how your battery is actually going to behave and some of the unique characteristics of that battery. So this data, right, it helps tell us things like what's the immediate, performance capability of the system. That's pretty obvious, right? It can tell you things like device chemistry, right? This is kind of what we talked about last time with DQDV, right? You're actually able to understand a lot of what's actually happening within the system, right? By kind of diving in and understanding and doing some of these data processes and transforms, right? To help you see these peaks and valleys, right? That may come from your normal, constant voltage charging steps. So that's one piece, right? Another piece is it kind of gives you what I'll call indications of battery health. And we'll talk about that a little bit more here in just a second. And it also gives you indications about critical system problems. And I kind of define systems as specific components or fundamental characteristics or anything going on within that battery, okay? So these are things that this battery data does. It gives us all of this information. What's not immediately clear without what I'll say is well-designed testing, okay? So you can do really well-designed testing and you can get to these sorts of things, but you have to have the data analysis and you have to have the good, well-designed test. Once you have those things, you can do cycle life prediction, you can do mechanistic understanding, you can do all of those things. But if I just get one curve of data, right, I can't tell you how long it's going to last, right? I can't tell you exactly what's going on in that system. I have to have well-designed test protocols and testing to be able to then apply my analysis to really help get towards these things. And that's going to be, some of what we'll talk about today. So let's talk about your raw data, right? So here I'm just applying looking at a battery. It's obviously what I've shown there is just a battery that's being charged at constant current. You could add a constant voltage step if you wanted to. That's not really what I'm concerned about right now. What I'm concerned about is that you take this data, right? And kind of standard insight tells us that you're going to take this data and you're going to do things like capacity, coulombic efficiency, maybe generic DCIR, right? There's a few things, right, that people really look at. We talk to a lot of people around the battery industry, and these are some of the major things that people are looking at. And this is what I like to call standard insight, right? Capacity, coulombic efficiency, generic DCIR. These are things that most people are taking a look at. So if you kind of want to look at this a little bit deeper, right? Like, what is standard insight? All right, well, people are doing capacity calculations, right? They take their charge capacity, and some of this may be really basic to you. Some people, this may be more newer information, right? But we have this charge capacity right here. And charge capacity is basically the amount of time that passed times the current over this entire voltage range, right? We have a starting point, and then we have an upper cutoff voltage, amount of capacity that passes here, right? This is going to be what we'll call our charge capacity. Then you go from here, you start a discharge, and you go all the way down to your lower cutoff. The amount of time that passes there times the current that you applied up here is going to give you your discharge capacity. Similarly, we can do coulombic efficiency calculations then. So we could take this discharge capacity that we had, and we could divide it by the charge capacity right here. And this is going to give us what we call coulombic efficiency. And Stephen will talk about this a little bit more. Probably if you were working in the battery industry, these two things are very, very familiar to you, right? And then we also have generic DCIR, right? Which is basically, we're going to look at some voltage jump here, right? A lot of times people apply a current step. And during this current step, they basically select a point below where the current was applied before the current perturbation, and then sometime after the current perturbation. And then they basically do V equals IR, and they get the internal resistance. The direct current internal resistance is what DCIR stands for. And they kind of track that. So these are three really important metrics and kind of what people are already looking at, right? And Voltaiq helps automate the calculations of these things, but also provides you so much more insight, right? And I think that that's kind of where I want to head from this point moving on, is that there's a lot more than we can do than just understanding capacity and just generic coulombic efficiency and generic DCIR. There's a lot more that can be done. And part of the process that we were talking about is, how do we automate that? How do we make that as streamlined and as simple as possible? How do we improve that so that the data that you're looking at is really going to help drive you towards business insight? Okay. So this is standard insight. Kind of the next thing here is that there's a lot more that we can do with that data, right? And we talked a little bit about this last time, differential capacity and differential voltage, or DQDV, right? We talked about that. That's one thing that I would say is critical insight. I'll talk a little bit about rest steps here for just a few moments. And then, Stephen will talk about UHPC and coulometry. But, a lot of people are really focused kind of on this black section of this graph here, the kind of standard insight. And we really want to try and help encourage people towards critical insight, because that's going to get you towards that mechanistic insight sooner. And it's really going to help you, eliminate problems, improve your product, and get towards profitability as quickly as possible. So let's talk a little bit about rest steps, right? This was we talked about DQDV last time. Let's talk a little bit about rest steps this time, right? So rest steps and performing really great analysis on rest steps is a really, really great way to get towards mechanistic insight as quickly as possible. So I have a current perturbation here. This is just a nice charge, constant current charge, followed by a rest. Now, this could be in some sort of pulse, right? But it doesn't have to be. This could just be after, your normal, charging cycle, right? Or this could be after a series of pulses. It doesn't really matter. But basically what we're going to do is we're going to analyze the rests, right? A lot of times when people talk about DCIR, they talk about investigating the leading edge of a pulse. And now I'm going to talk about actually looking at the trailing edge. When you've actually removed all of your current, what's the information you can get out of this system? So one of the things is you can look at what I say is the difference in voltage here between 0.4 and 0.3. And this is going to give you information about the internal resistance of your system and the kinetics. And there's ways that you can actually separate these out. So you can actually just be studying the kinetics. So this is a really, really important point. The next part is the difference between what I'll say is 0.5 here and 0.4. This is actually going to give you transport information. And we'll talk about all this in just a second. And then kind of, if you want to look and compare what happened over the entire pulse from equilibrium point till after the pulse happens to the next equilibrium point, this is going to give you information about thermodynamics. So just by looking at one single rest step, you can get information about transport, kinetics, and thermodynamics. So this is a really, really powerful tool. So let's talk just briefly, when you're analyzing this rest step, what's something that you can do to try and extract information about your kinetics? So right as that, the current is removed, there's going to be a pretty immediate instantaneous drop that is going to happen to your voltage. The reason that this instantaneous drop happens is because you are removing the current. And so there was a buildup of charge that was associated with actually driving what we'll call charge transfer, or basically a lithium ion going from your electrolyte into your electrode, or going from your electrode into your electrolyte. There was a buildup of overpotential, what we call, but basically it was a voltage that had to be sacrificed in order to actually make the reaction happen. As soon as you remove that current, that goes away, right? And so this right here, we could kind of look at an EIS measurement here to help us identify like what time we should be looking at this data point right here. So what's the time difference between here and here to help me extract this kinetic information? An EIS is actually a really helpful way. Just a representative EIS is actually really, really similar. If you have very similar chemistries, basically the same frequency, right, or the same amount of time is going to apply to pretty much all of those systems, right? So from this data point down to this data point right here, this is going to give us information about the overpotential that had to be applied to actually drive our reaction. That's really, really helpful information to know. Gives you a lot of insight towards mechanism, right? Then from that data point all the way towards the end until you reach equilibrium, right? When you had applied your current, you would kind of set up this concentration gradient across your cell, right? It was going from cathode to anode, so you had a concentration buildup across your electrolyte, but then you also have a concentration buildup at your individual electrode particles. So if you're putting lithium into your cathode, right, or into your anode, let's say, right, you're going to have a very high concentration point at the surface of your anode, and then you're going to have less of a concentration further into the middle of that particle, right? And so over time, everything's going to want to reach equilibrium, and all those concentrations are going to want to go back towards some sort of steady state condition, right? And as time progresses, this voltage is going to decay, right? And if we track that voltage decay, we actually get a lot of really important information into the transport phenomena that are happening within your system. So these are things that Voltaiq can automatically calculate for you, right? So these are important things that help you understand that critical insight, help get you towards mechanistic insight, help get you to the bottom of the iceberg as quickly as possible. And when you get to the bottom of the iceberg, right, you know cycle life, you know capacity, you know your SOPs, you know your materials for quality, you know all of these things, right? You now have a full picture of the iceberg, right? So that you're not going to be like the Titanic and gash a hole in the side of your company because you didn't fully understand what was happening underneath the surface, right? So that's really kind of what we want to try and get to. And, these automated calculations, this advanced analysis, right? The stuff that Stephen's going to talk about here in a second with UHPC are really, really helpful tools at helping you get the full picture of that iceberg. All right, I will pass it off to Stephen and he will take it from here.
All right. Thank you, Kevin. All right. So continuing where Kevin left off here, we're going to talk really, I guess, diving into specifics of data, specific case studies that we've done actually internally at NOVONIX. So these are real experiments, real case studies that we've done internally on our own research streams. So hopefully everyone here learns a little bit. Everything I'm going to go over is not always specific to, using our equipment. So I think everyone here can still learn something, even if you don't have NOVONIX UHPC, or even find ways that NOVONIX UHPC would be useful within your company. So I'm going to give a quick introduction to NOVONIX, then talk a little bit about the topic today: qualification, validation, development, similarities, differences between those things, and then do a little bit of a recap of the theory around UHPC. What are we really measuring? Why does it matter? And then jump into these case studies. Again, all real world case studies done on our pilot line with real cells and data, and hopefully teach you a little bit about how you can use these techniques or this equipment in your own lab. So a quick overview of NOVONIX in general, just to give some context of why we have a lot of this information. So starting right here in the middle, we've got our NOVONIX Battery Technologies Solutions Division. This is where I am right now in Halifax, Nova Scotia in Canada. And this is where everything kind of started just over 10 years ago in Jeff Don's lab, developing ultra-high precision coulometry cell testing equipment. And, from there, we branched off. We built up a in-house pilot line for cell building and cell testing. We have over 3,000 channels in-house of various cell testing equipment, not just our own. And then started branching even further into the anode material space, the cathode material space, really become a leader in anode and cathode materials within North America with offtake agreements with the likes of Panasonic, PowerCo, and Stellantis. So how we got all these places, really, we owe it back to our roots, using ultra high precision coulometry data, using those techniques to really make leaps and bounds through the industry and get places much faster. And that's really a big theme right now across the whole battery supply chain is really focusing this year and getting places faster, reducing scrap, just getting online, getting profitable, as Kevin said. And so NOVONIX and Voltaiq both really focus in this space, helping our customers, partners get there. And of course, I'm going to be talking about NOVONIX UHPC. So this is the highest current and voltage accuracy precision equipment available for electrochemical cell testing. This is equipment we make and design in Halifax, Nova Scotia, and sell globally to tier 1 manufacturers, to startup companies, to academic labs. So I'm going to just go into talking about these three things really quickly to give a little bit of context around some of the case studies I'm going to talk about today and why they're relevant. So we've got qualification, validation, and development. There's some similarities here. Qualification and validation focus on meeting existing standards, and development emphasizes it's more on the innovation, the invention, the creativity. So qualification, we're talking about things like equipment, materials, and systems, proving that they're fit for use. So this could be you're trying to qualify a new vendor for cathode materials. Validation can be confirming processes or methods consistently meet standards. So potentially validating over and over again that a new mixing procedure is always going to consistently meet spec. And then development. So of course, this is that innovation side, creating new, improving products, processes, or technologies. And of course, there's sometimes a logical flow between the three of these, and sometimes people work in completely parallel work streams with them. And I think the common thing for everybody in this call, everyone across the battery supply chain right now, industry conditions are making it really important to have really high confidence in a very short amount of time. And what I really want to hone in on today is not necessarily all kinds of testing techniques that everyone might be familiar with, but trying to identify gaps in those existing pieces of equipment or techniques that we can really help with, that UHPC can be injected into a lab and really fill those gaps and get those conclusions faster, get those mechanistic insights that Kevin was talking about. And of course, when paired with something like a software like Voltaiq, this type of equipment can really get you these automated insights very quickly. So I'm going to go back and talk about these gaps and where UHPC really fills in. So existing cell testing equipment that everyone's probably familiar with, there's, a handful of brands and kind of spec levels that almost exist in everybody's lab. And these really excel at things like testing cells to end of life, rate capability, DCIR, GITT, application and drive cycle simulation. These applications where you're getting these bulk metrics where, small errors don't really matter. If you test 1,000 cycles to 80% or 1,050 cycles to 80%, it's not really going to matter too much. If there's a few decimals of a percent difference between your 2C discharge capability, it's not really the end of the world. These are things where the errors in your system are kind of at the same level or smaller than the errors within your tester. And a lot of lab-based high precision testers are, say, 0.05% precise. And where these gaps really exist are understanding impacts on things like lifetime early. We're trying to do hours or weeks of testing instead of months or years, or things like tiny issues in cells before they become big issues. So this can be QA, QC, looking at small, tiny amounts of self-discharge that may be, completely within the noise of the current measurement of your recycling system. And these are, this common theme, or these were bulk metrics of which, small errors matter a lot. And so what I really want to emphasize here is that, UHPC can do almost everything that these of cell testing equipment can do, but we've really designed it, and the thought process behind developing it is to detect and elucidate these small differences that matter early. So that's really what I'm going to focus on today, is small things with big implications. So in order to get into these case studies, I'm going to do a little bit of a recap. I'll go quickly through it of some of the theory that I discussed in our first webinar. So if you're not familiar with it, it's on YouTube right now, and you can go and check it out and maybe go through it in a little more detail. So Kevin already discussed, what we're doing when we're charging a cell, discharging it, getting these metrics, integrating time and current to get capacities, et cetera. And what I always like to think about is like, what are we actually measuring when we're measuring something like capacity or current? And really what we're making a direct measurement of is two things: the potential across the two electrodes. And we're not actually measuring, say, the amount of lithium that we're moving back and forth when we're measuring current. The only thing we're directly measuring up here where this tester is, this diagram, is the number of electrons through the external circuit. The more accurately we can do that, the more we can figure out what's actually going on in here. So we're really not directly measuring this lithium moving back and forth. So in the upcoming diagrams, I'm just going to focus on what charge looks like and all the other mechanisms that happen in here and what it does, what it kind of affects with this measurement that we're making. So I think everyone's fairly familiar with capacity loss. These are things like SEI growth, active material loss, impedance growth, that as we're moving lithium across in the cell, we're counting these electrons. We lose some of these due to these failure mechanisms, due to electrolyte reacting at the anode material, or active material loss or cracking happening at either electrode. And cycle to cycle, we end up losing some capacity here. And so, of course, the more accurately we can track those electrons back and forth, the more accurately we can see how fast something is fading and not, lose any insight due to the noise of our cycling system. But these aren't the only failure mechanisms happening in a cell. They're not the only thing that's contributing to counting electrons through that external circuit. There's all these other types of mechanisms occurring that can be due to electron donating reactions. This is something like electrolyte oxidation or shuttle mechanisms, transition metal dissolution, and even physical self-discharge or shorting. So looking at this diagram here, we have things like transition metal dissolution, where we're shuttling charge across the cell, and to create charge balance, we actually have to pass current through the external circuit, and we're counting these electrons. With a short circuit, the same thing is happening. As we're trying to charge the cell, there's this internal self-discharge, and we're counting all of these excess electrons. This shuttle mechanism acts the same way. We're shuttling electrons across the cell and counting them back as we're trying to continue charging the cell. And then even these irreversible electrolyte oxidation reactions. So we've actually got all these other contributions. And in each of these cases, when we're talking about all these excess electrons we're capturing, none of these processes directly cause capacity fade. So these are things that you wouldn't actually catch with a low precision piece of equipment if you couldn't capture these electrons accurately enough. So if we can capture, the electrons we're losing due to capacity fade mechanisms, and then all these additional things, which in traditional cycling equipment, I'll show later, is actually kind of invisible mechanisms, because the rate at which these happen are so small. We can get really interesting mechanistic insights into active material, electrolyte development selection, lifetime prediction modeling. You could see how some of these insights, if you can really accurately capture them, could really improve lifetime prediction. And especially things like quality control, finding these tiny, tiny shorts really early in process development. And we can measure these things a few different ways, and I'll show a few of them in this webinar. Leakage current, just direct measurements of say, these shuttling mechanisms happening. Open circuit voltage drop, which is more of kind of a traditional way of measuring some of this, the self-discharge occurring. And then charge endpoint capacity slippage as a cycle metric, which is a cycle metric that's typically not used very much or talked about, because it is really only possible with ultra high precision equipment. And I'll just show this for reference. This is how we calculate this charge endpoint capacity slippage. When we're charging and discharging a cell multiple times and looking at a cumulative capacity, so instead of resetting the capacity to 0 every cycle, we just pick up and keep counting where our discharge ends off. So as we keep cycling, charging, and discharging, some of these excess electrons we count during charge end up making us walk a little bit further during charge than, say, the theoretical capacity of that cell. And then during discharge, we don't come all the way back to 0. So then the next charge, we walk back a little further, and we end up with this slowly walking voltage curve out to higher and higher cumulative capacity. And if we just track this endpoint, we put a pin in the map here, and each cycle we track where that endpoint is, we get this plot of this charge endpoint capacity slippage. And with very high accuracy and precision cell testing equipment, this is a very consistent cycle metric. You can see here, these are pair cells made on our pilot line with 2 different electrolytes. One of these, electrolyte 1, just has vinylene carbonate. Electrolyte 2 has vinylene carbonate and propane sultone. And that propane sultone really starts to shut down these high voltage reactions at the cathode and mitigate this charge endpoint capacity slippage. What's likely happening here is we've got a lot of this electrolyte oxidation happening, counting a lot of these excess electrons, which drive our endpoint to higher and higher capacities. And we can also look at this metric as charge endpoint capacity slippage per cycle. And this is more like the rate of which these reactions are happening. And again, we can see electrolyte 1 is a much higher rate of these mechanisms happening than electrolyte 2. And this gives us these like very quantitative insights into things that we can't really see with other types of cycling equipment. Here I'm showing, what this all really looks like when we go into the full picture. And we like to talk about coulombic efficiency here because all of these mechanisms going on affect the coulombic efficiency. And we've got a couple different terms to that. One has to do with all those capacity fade mechanisms, and the other has to do with all these excess electrons that we're counting during charge that act as self-discharge when we're in a discharge step that we were talking about in terms of that slippage term. And if you can measure coulombic efficiency very accurately, it means that you can also split these things out and look at fade independently of slippage, because really these two things contribute to your coulombic efficiency. The mismatch between your charge and discharge has contributions from both of these. So in all of these case studies, we're going to look at one, the other, or both, and try to make these insights not just with coulombic efficiency, but really get to the underlying mechanisms quicker, because we have the required accuracy to do it. So I'll show quickly how we do this, fundamentally going all the way back to the raw data and then the cycle metrics that we calculate. So here's a slide that's just showing 3 other types of cyclers that are commonly used in everybody's lab right now. And then what this looks like when you put on 8 or 9 identical tier 1 cells. So this is tier 1 18650s, all put on test and cycled 20 times. And this is at a low rate. And this is really key for doing really accurate coulombic efficiency measurements. Kevin was talking a lot about kinetics. And in a lot of the case studies I'll show here, but not all, we do a lot of slow cycling. We try to remove the kinetics and focus on the electrochemistry and figure out what's happening at an electrochemical stability level, and then try to understand how that's contributing to fade or slippage. So this test is slow cycling at about C/15. And you can see the spread in these, identical cells on each of these different types of cyclers. And then it's actually showing up in green here. This is supposed to be black, I apologize. Whereas these 8 or 9 cells all have almost the exact same coulombic efficiency being measured on the NOVONIX system, whereas everything else is 1 or 2 orders of magnitude in uncertainty larger. So we can really kind of blow this up to see the spread of these cells on the NOVONIX UHPC system. So what this really means is if we make a very small change now to that cell's chemistry, to the electrolyte, to the cathode material, or even have a cell that has slightly higher self-discharge than another cell, but on the orders of or tens of ppm, we can actually measure that, identify it very early in a matter of just a cycle or two, and make really insightful conclusions or identify outliers. And again, you can compare this, or you could use this for any of qualification, validation, development, just depending on what your use case is. The way we do this is very high precision and accuracy voltage and current measurement and sourcing. So you can see those other cyclers, there's variations in their accuracy, in their precision. You can see the bit switching in some of these, whereas we just try to be at least an order and usually 2 or more orders of magnitude tighter in all of these categories of voltage and current, precision and accuracy. So I won't get too much into those details, and I'll just show here how these translate into these cycle metrics. So we showed coulombic efficiency in the previous slides, where some of these have good precision, but poor accuracy and poor precision. And then what this really means for some of these other cycle metrics, capacity fade, eventually all of these cyclers can show you the trend eventually. This is why anything can do these long-term cycle aging experiments where you just put a cell on and 1,000 cycles later, all of them will tell you more or less the same thing, how much capacity is left at that number of cycles. But then when we look at things like charge endpoint capacity slippage, some of that really insightful information on some of these more or less invisible metrics with these traditional cyclers, you can see this endpoint slippage on all of these identical cells. Some of them are walking backwards in capacity, in cumulative capacity. Some of them are walking forwards. And really, you need this extra couple orders of magnitude in accuracy and precision with our cyclers in order to measure this charge endpoint capacity slippage, because it's just not a good metric on any other type of equipment. And really, this is because those rates of reactions are happening at a magnitude that's less than the noise of the measurement in these pieces of equipment. And we can see that just in the current here. So without further ado, I'm going to get into the meat of this, into these case studies, which is what everyone's probably more interested in, and some of the theory behind it. So as I said, NOVONIX is a battery materials company. We have a ton of work streams in cathode and anode development. And so this case study here is focusing on cathode. So what's really interesting about trying to interpret some of these data is it really doesn't matter whether we're looking at precursor selection or coating or dopant development, process or recipe optimization, or vendor selection of a final cathode material. This case study is relevant to all of these things, and it's just what we're trying to get out of the data and how we're trying to interpret the data that matters. So in this, we're just going to talk about cathode 1, 2, and 3, but we can all use our imagination and pick one of these that's most relevant to us to make our conclusion at the end. So the goal here is, in this case study, is to screen cathodes with different properties, find the most electrochemically stable material for this application, and try to make a decision in less than a week on this. So there's a few methods that we employ here. So point cell UHPC measurements of parasitic current in voltage conditions. So this is doing like a direct measurement of the current during a voltage hold, which will give us this parasitic current. Full cell cycling to measure UHPC cycle metrics. And then we're going to compare this to cycle aging. And we're going to do this in our in-house pilot line with the same anode, same electrolyte test conditions, all cells filled on the same day, tested by the same person with the same batch of electrolyte, et cetera. So we're controlling all these different parameters so we can really say any differences that happen here have to be coming from this change in the cathode that we're making. So here we're showing the data during coin cell voltage holds. So we're doing a slow cycle here and then just doing a voltage hold at 4.4 volts and then trying to measure directly the parasitic current during this hold. And so the measurement of the current is really important to screen the cathode, because ultimately once all the kinetics kind of level out, all you're measuring is the amount of electrons that you need to be kind of shuttling through that external circuit in order to maintain that state of charge. And this is that slippage mechanism. And our UHPC measurement here is less than, the accuracy and precision is all less than 10 nanoamps. I'm looking here at hundreds of microamps. So Any of the noise that we're seeing here is actually like electrochemical system level noise, maybe small fluctuations in temperatures. So all of this is real measurements occurring. And so we can take this measurement directly and start to make conclusions about the electrochemical stability of these cells. And I think there might be a small typo here, but cathode C here, the black, has the least parasitic current that we see. And then cathode B, actually, the blue here has the largest. So the hypothesis here is that in cycle aging, we'd probably see that cathode B is failing first. And you'll notice this is just within a couple days of starting these tests. Moving these into full cells, and this starts to get a little more complicated. We're not directly just measuring the cathode. We're looking at fade happening, lithium inventory loss into the anode. What's really interesting here is we look at these different metrics, the Coulombic efficiency, and we see that cathode B, which had the highest parasitic current, is the lowest Coulombic efficiency, and then cathode A, and then cathode C. So this follows that direct parasitic current measurement very well. And what's interesting here is when we break this down into the two constituents or two main contributors to Coulombic efficiency, we see the discharge capacity fade and the charge endpoint capacity slippage. And really, these are shown on the same axis to demonstrate that the main driver between the differences in Coulombic efficiency is not how quickly the cells are losing capacity. It's actually these other more invisible mechanisms like electrolyte oxidation at the cathode, like transition metal dissolution from the cathode. These electron donating mechanisms that are decreasing our Coulombic efficiency, not what is making the cell fade faster. And we can see that when we go into looking at our long-term cycle aging out to a couple thousand cycles, right at the start, everything does actually fade very similarly. And, we see that in our just a couple weeks of UHPC cycling too. But really what ends up making these cells start to fail, to roll over, to maybe dry out of certain electrolyte components are these slippage-based reactions, not directly fade-based reactions that we see at the beginning of life. This black and green look very similar until about cycle maybe 600 or so. So these UHPC measurements can predict this order of failure due to this cathode electrolyte instability very quickly. And it really can tell us, once we can separate it into these two components of Coulombic efficiency, where the failure is coming from. And this becomes a lot more important later on when we're not just looking at cells where we're controlling everything about the cell and making very good apples to apples comparisons, when we're looking at very different cells. And we'll get to that when we start talking about lifetime prediction and generalizable lifetime prediction. So here's another cathode project, which is more or less the same as the last one. It's just showing, like, even later in life, how very, very similar materials can perform very, very differently. The small differences that really matter later on. So in this experiment, we had two different cathode variations tested in otherwise identical cells, again, from our pilot line. We did cycle aging at 25 degrees and UHPC at 40 degrees, again, doing this UHPC cycling very slowly in order to just get the differences in electrochemical stability. And we see that the results of the cycle aging, which I'm showing discharge capacity and this delta V, the difference between the average charge and discharge voltage, look very, very similar until about 1800 cycles. So this is pretty long into cycling. They had exactly the same delta V or voltage hysteresis. They had exactly the same discharge capacity fade. Everything looked, really good for both until months and months into cycling. Some disassembly showed some lithium plating, localized electrolyte dry out. And so clearly something's happening that really must originate at the cathode, because all these cells were made and tested the same way at the same time. So our hypothesis is that UHPC data should identify this relatively quickly. And when we dive in, again, very much the same as the last experiment, we see that very early on, we've got the NMC 622, number 2, has a much higher Coulombic efficiency than number 1. And this alone isn't enough to tell us why the cell failed. But if we break that up into the fade and the slippage base terms, we can see the fade is almost exactly the same. We're losing capacity, we're losing lithium inventory, at almost the same rate in both of the cells. But really what's going on are these electron donating reactions, these slippage-based mechanisms, which are happening at twice the rate in number one than number two. And so these are really big differences in UHPC that would otherwise be in the noise of other types of cyclers. So it's really key is to know that just in a couple days, like this trend doesn't change. In just a couple cycles, we would have been able to make the conclusion this cell has a lot more charge endpoint capacity slippage, and it's likely going to fail sooner. And again, this doesn't show up for months and months into normal cycling when we can see it in days with UHPC. So moving into the other side of our business, into graphite. So we do a lot of different experiments with synthetic graphite, comparing it to different methods through validating new precursors to developing new coating methods, all kinds of different work streams. And so our goal is always to screen these materials for electrolyte reactivity using UHPC. Again, things like rate capability are a very important part of all of these work streams, but other cyclers really excel at those. So what we want to focus on using UHPC for is diving in to those really tiny mechanisms very accurately, getting a really nice quantitative metric or ranking of these things, and carry that forward. So we're looking at screening these materials for electrolyte reactivity, and we're going to do this first, just like we do a cathode in coin cells. And you can see that compared to other high precision testers, there's both an improvement in the accuracy and the precision of the data that you can get with these voltage hold style tests, where you're directly measuring after all of the kinetics kind of even out the parasitic current happening, the rate of those reactions. We can then use those rankings to look at, this is the endpoint here of these tests, make some kind of ranking, look at other types of metadata, such as the surface area, such as the different process conditions that may be different with each of these experiments or precursors, et cetera. There's a lot of different axes to look through. And that's another thing that we're going to be discussing in the next webinar too, is diving kind of into the metadata and tracking some of these processes and parameters to make some of these kind of deeper insights, especially within Voltaiq, which allows us to track all of this, other metadata and make some of these conclusions. So another case study here, very similar, again to cathode, is doing graphite screening with full cells. So again, cells made on our pilot line, all the same cathode, all the same electrolyte, and we're just varying different types of anode. And here what's really interesting is we almost see the flip side of what we saw with cathode, and this is due to where these mechanisms typically occur, where we see these big differences in coulombic efficiency. And again, these axes are really blown up on other types of equipment. All of this might just be within the noise of the other data sets. We see these really consistent differences in coulombic efficiencies, cell to cell. And when we break this out into the fade and the slippage term, like I said, we see that almost a flip of what we saw in the cathode, where in cathode we were driving these differences with charge endpoint capacity slippage, and we saw basically the same capacity fade rates. But here, we're seeing this difference in coulombic efficiency is actually driven by the difference in the fade rates of the cells. This is due to losing lithium inventory into SEI growth or active material loss, because we're fixing what's happening at the cathode and changing what we're doing at the anode. So we're seeing a lot of these differences come up and fade. Whereas for most of these materials, we see almost the exact same slippage. These electron donating reactions that typically originate at the cathode are the same in each of these cells. We do see a very consistent outlier here. Those pair cells are quite consistent, and this slippage is a little higher. And this kind of points to there's something else happening in these cells where, maybe there's a mechanism happening at the anode, which is generating some type of crosstalk mechanism, something that ends up being unstable at the cathode, but actually originates due to some SEI byproduct. So, this kind of warrants a little bit more looking into and highlights that there's something different about this material, and we can actually see where these things are happening. And that can help us dive in, do more analytical experiments, and really look into, what is different about this natural graphite? Is it the way it was coated? Does it have a very, very different surface area? Allows us to dive in and understand what's causing this to happen. And then, of course, this early ranking that we can get, and just, again, this is about a couple weeks of cycling, but you can just look at a few cycles in a matter of days and then predict this order of performance that's going to happen months down the road in normal cycling on a traditional piece of equipment. So this really allows us to get places much faster and not just necessarily predict, a ranking of how something's going to perform. There's a lot of insight here that may actually allow us to quantitatively predict things. And that's where we're going to get by the end of the webinar, is more quantitative predictions. So I'm going to kind of break from material work and get into more application-based development. So looking at cell orientation, this is a case study that I think is really interesting because it diverges a little bit from the traditional UHPC style tests that are published in the literature that typically, and the ones that I've shown earlier, that are done slowly cycling. And it actually does different rates, actual like application rates, C/3, 1C, and looks at the effect of that on the performance of the cell and application. So this is a really neat paper that was published by Connor Aiken, who's actually in our group at NOVONIX. And this was published during his PhD. And we continued a little bit of this work because it was fairly interesting. And we thought there was a way that UHPC might be able to contribute. And essentially what they were looking at is electrolyte pumping. What happens when you're testing a cell, especially in a rigid container, like a cylindrical cell, and you're doing charge and discharge, your materials are swelling and contracting. What happens to electrolyte? Where does it go? And so they built an interesting device that they called the jiggler, which oscillates a cell back and forth and looks at the moment of inertia of the cell to figure out where the electrolyte is. And it was a really neat paper. I really recommend it for anyone thinking about how cells perform and what goes on inside of them, not just electrochemically, but physically what's happening. And since then, there's been a lot of work done in this space and a few cool publications. So we really thought, well, this work was done with taking cells in a horizontal orientation, and a lot of cell testing is done where you would fixture, say, a cylindrical cell horizontally on a board and put it into a cell cycling device. But then even in the same lab, you may have a chamber that holds cells vertically. And especially when we think about this moving of electrolyte in and out of the jelly roll, pumping electrolyte, does that have an effect on cell lifetime? If I test my cell vertically versus horizontally, am I going to get different pieces of information? And so we did this experiment in-house on commercial 21700 cells. And what we did is we tested them with different rates. So C/3 symmetric charge discharge, or a C/5 1C charge discharge. And what this is trying to do is keep the same cycle time, total cycle time, of approximately 6 hours, to kind of control that side of the kinetics, and then change how much time the electrolyte has to really, get back into the whole jelly roll, distribute, and have a really nice, even concentration gradient within the cell. And just doing some cycling on a low resolution cycler, we see over hundreds of cycles, really no difference between the two. And then really during this like faster discharge, we start to see some divergence when we do this 1C discharge, and maybe not allow all the electrolyte to get back in to the jelly roll during discharge before charging again. Whereas the difference isn't as big in the C/3. So we said, okay, this takes hundreds of cycles to be able to tell, but obviously something fundamental is going on, and probably very early. Can we see this with UHPC? So we did the exact same protocol. So this time not trying to remove kinetics, this time trying to see the effects of kinetics on cyclometrics. And so we did C/3 cycling for only 75 cycles. And this is only, this is days of cycling, maybe about a week on UHPC, and then C/5 1C. And right away, you can see these dramatic differences between the two with coulombic efficiency between the horizontal and vertical. So We've got kind of identical Coulombic efficiency with C/3 between vertical and horizontally oriented cells. And then C/5 1C, we've got actually very poor performance relatively in these horizontal cells and fairly inconsistent. So this is a bit of a red flag. Something's going on very early, almost in the first couple cycles, within hours of starting or a day of starting this test. We can see this big difference. And we already know that the capacity fade is the same between these two cases, vertical and horizontal in each of these cases, because that's what we saw early in the first few hundred cycles. So we're not even going to look at capacity fade here. We're just going to look straight to charge endpoint capacity slippage. And this is really interesting that this Coulombic efficiency between the vertical and horizontal cells is driving a difference in charge endpoint capacity slippage. So these horizontal cells with this faster discharge are experiencing more of these slippage-based mechanisms somehow. And this is actually leading eventually to this worse performance and this divergence later on that takes hundreds of cycles to find out. And there's been some interesting work that's actually done by a group associated with BMW that has identified this as this electrolyte movement caused by this pumping causes really huge concentration gradients in the salt and salt depletion during fast cycling. And so if we kind of picture a cell vertically, we've got this nice, almost like capillary action of electrolytes to be able to get back into the jelly roll as a cell is resting, especially as we, go to a faster discharge and not necessarily give as much time as the cell is, has pumped out the electrolyte during charge to come back in, not giving it very much time. The vertical oriented cells, it can travel back into the jelly roll a lot more uniformly. Whereas when we turn the cell on its side, electrolyte doesn't have to just get back into the jelly roll. It also has to get around all of the lines and back into the top of the jelly roll as well in that orientation. And so what you end up with is very big, very dramatic salt depletion and concentration gradients depending on how you orient your cell. And this can preferentially lead to oxidation of electrolyte energetically and increasing this charge endpoint capacity slippage. And this is really important when, kind of choosing your application or even when testing cells and trying to analyze the results of those cells. If your application is vertically oriented cells and you're testing horizontally, you might be led, down the wrong path in your cell selection or in BMS condition selections by this. And just within a couple of days, we're able to identify this very quickly with UHPC. And so there's just some pictures here of some renderings of packs where we've got the Tesla pack with cells that are vertical, and then another rendering of a pack here where there are horizontal based cells. Another quick case study I'll go over is relevant again for cylindrical cells. Really, it can be for any cells. This was a study that we did when we were trying to optimize filling and wetting conditions of a certain spec of 18650. So when we've got cells in a very constrained environment, especially as we're trying to increase energy density and get to manufacturing, it's really critical to validate all these process steps and even optimize how to get there faster and how to get there more consistently. So this is a case study where we were trying to figure out optimal conditions for vacuum staging during filling. So we would do multiple injections of electrolyte into 18650s and then do different vacuum staging procedures before crimping them and putting them on formation. And the big challenge here is, a long development validation time. How can we do this as quick as possible to get the good answer on how consistently we're wetting the cells? Are we fully wetting the cells? And developing the testing procedures around that. Do we cycle the cell for 100 cycles, 1,000 cycles? How do we really know? And what we actually found is if we did our formation procedure, which this is just a standard formation procedure that we do on a lot of cells here, during this what we call a wetting step here, where we do a 1.5 volt voltage hold, we found that if we really looked closely at the current during this voltage hold, with UHPC, we were able to see at very small current levels, differences in the spread of the current. And this gave us really good insight into how much material was still needing to be accessed and wetted at a certain amount of time into the wetting procedure. And this correlated really well later down the line with the consistency in the cell capacity, the performance and rate testing, and in cycle life. And really, we were able to do that with very precise current measurements just in a couple hours. Within a day, move on to the next set of conditions without having to then put those cells on test to wait for those measurements. Once we confirmed that we could, really do high throughput experiments just in a day, we really honed in on using UHPC during this formation step to get to our optimal conditions faster. So getting into the last two case studies now, this one might be relevant to folks on the call, looking at formation optimization. So, formation is kind of a secret sauce in the industry. And depending on your cell chemistry, your electrolyte, your application, it could be done so many different ways with different storage steps or aging steps or rates and conditions. So this was a really interesting project that we did with an automotive partner. And, what we did is we took a single chemistry, this is LFP graphite, and took over 100 pouch cells. And these are 2 amp hour pouch cells, and varied all the formation procedures. And so this was varying different temperatures for storage, aging SOCs, et cetera. And then took the cells and then moved them into UHPC cycling for a couple of weeks and cycle aging until they reached 80% state of charge. And this is a correlation matrix here that is dimensionally reducing the data and looking at individual formation metrics such as for efficiency, discharge capacity, charge capacity, the UHPC metrics such as the final average coulombic efficiency or the final slippage value during the UHPC testing. And then the cycle aging parameters like number of cycles to 80%. And looking at all of these metrics and how they correlate with each other, we can see that there's some very strong correlations between the UHPC cycling metrics and how the cells performed in cycle aging. And I'll zoom in on this in just a second. I just wanted to illustrate, there's a few of these correlations in other places, but it's very strong between cycle aging and UHPC. So this gives us a good gut check that we can use UHPC to quantitatively predict some of these influences on cycle aging. So I'm going to jump just into one of those squares, that we see some good correlations down here. And this might be one of the more obvious ones that we've talked a lot about, looking at something like coulombic efficiency and how it relates to the cycle life to 80% here. And seeing here that there's actually a really nice correlation that is really quantifiable. We could actually use this and many other axes in this experiment, such as the time of the formation or the cost of the formation, to really optimize what gives us the best cycle life with, say, the cheapest formation path or the quickest formation path. And to drive home why this is really possible with UHPC, I'm just going to put on the error bars of the coulombic efficiency measurements that I showed earlier of these other cyclers these are the brands that are very common in labs across North America. So the error bar on UHPC being, about as big as one of these points, we're really going to be able to take, say, the next round of this optimization, do the UHPC testing, find this final coulombic efficiency or fade or slippage value, and correlate directly to where that's going to land in cycle life and determine if it's even worth testing that cell. So saving a lot of channels in the lab and time to validate new procedures. So this is kind of all foreshadowing that these next steps here is really like, if we can make these really nice clean models, can we generalizably use UHPC to predict cycle life? And I showed this in the last webinar, and I'll show it again, just because it's a really nice set of data. It was a really nice collaboration, and it really shows the use of all of these now new high precision metrics that we have and how they can be used really quantitatively in any application. So this is a project that we did with a collaborator, Sandbox AQ. I think there's a couple Sandbox folks on the call today. And we worked with them, really bringing our UHPC testing, cell degradation, knowledge and expertise. And Sandbox brought their data pipelines, machine learning expertise. And we said, okay, how can we take UHPC and really use it to predict lifetime. The other great thing about NOVONIX is we have a lot of data, and that's really required to make these quantitative predictions. So this initial study that we're working on with Sandbox, or we finished with Sandbox, is using a controlled data set here, and we kind of show some of these parameters where we're really probing different types of failure mechanisms. We're not trying to like hone in on a very small space. We're really trying to generalize what's going on and challenge these models. So you can see this axis here of different temperatures going all the way from 25 up to 60, and the different voltage ranges or SOC ranges that we're testing in from 2.5 to 4.06 volts. So this is cutting off, this high voltage plateau of these iron nickel cell chemistries here. These are all 21700s or 18650s with a high nickel cathode and either a graphite or graphite silicon anode. And then doing this full range of discharge, 2.5 to 4.2 volts, and then 3.3 to 4.2 volts, and actually cutting off some of the capacity in some of these cells. So we're turning on and off silicon-based failure mechanisms, turning them up and down with temperature, and doing the same for the cathode side. And that makes it really hard for traditional methods of lifetime prediction or modeling to predict failure, because we get these things like rollover failure happen. In this work with Sandbox AQ, we were able to take standard model features, and these are all published features in works like the Severson paper on lifetime prediction, where we have things like delta QV voltage curve changes between various cycles, and then using discharge capacity trends early on in cycling to try to predict what's going to happen later. And really, there's not a great correlation between the predicted end of life and actual end of life. And this is a lot of the time in some of these overpredictive states, it's because of these rollover failures that happen. And if we work in UHPC model features, so this is using those same early features, just looking at the first 100 cycles here of the cycle aging data, and then layering in charge endpoint capacity slippage, fade, coulombic efficiency, all as features in a machine learning model, the model tightens up almost immediately because we catch some of these overprediction cases. We actually catch some of the underprediction with some of the capacity fade and impedance information. And we get really, really nicely performing lifetime prediction models. And again, this is fairly generalizable in this study because we're turning on and off these failure mechanisms. We're not testing every cell under the same condition. We're really kind of challenging this model to be able to do this accurate prediction. And today I've talked a lot about cycle metrics and only a little about raw data. And, there really is a lot of benefit to looking at raw data. We talked about DQDV, DVDQ in the last webinar. Kevin mentioned it a little today. And of course, if we have equipment with the highest precision and accuracy voltage and current measurements, we should be able to get really nice, clean DQDV. And that is something that UHPC is really good at. So this is showing some plots of DQDV or DVDQ, the inverse there. And on top of doing a lot of interesting qualitative fitting, now that we have really nice, clean peaks, we could also use these peaks, the locations, the magnitudes, how they move as features in machine learning models. So this is also something the team did. Is there's a representative chart here of a plot that shows the differences as we cycle a cell, the residuals of each cycle of DQDV. So you can see between cycles, say 5 and 6, the darkest line, there's some very tiny differences in the DQDV, which you can pick up very cleanly with UHPC data. And these just amplify over time into some very big differences. And these mean that these DQDV peaks are shifting and moving. This has to do with capacity fade, both at the anode and the cathode, depending on which peak we're looking at. And baked into all this raw data is all of that information about charge endpoint slippage and capacity fade, coulombic efficiency. So when we use this raw data and these features based on DQDV peaks, positions, magnitudes, and we use just that as inputs into lifetime prediction models, we actually get some really great performance in a very short amount of time. We can already see that cycles 5 and 6, this residual difference in this one cell, all of the features are there. They're just quite small. If we use only the cycle 5 and 6 difference versus the cycle 5 and 45 difference, the predictive behavior of the models is actually basically the same. So just in using the difference between a couple cycles, it's fairly miraculous. We're able to predict the lifetime of these cells in these very different conditions. So with anything that's, machine learning modeling based, there's a lot more work to do, a lot more generalizable data that we can add in. We have a lot more data here that we're working with our partners in and are going to, hopefully show a lot more of this in the future, showing a lot more generalizable data sets, different temperatures, rates, conditions, very different cell chemistries, and how UHPC is able to predict this lifetime. And this, of course, has implications in warranty estimations, in all aspects of development, qualification, validation. It just really depends on your goals and how you can use UHPC. So as a quick summary, just checking in with the 80 folks left on the call, based on where we started at the beginning of the webinar, how do you feel about the value of UHPC methods? Do you think it's most useful for qualification, validation, development everywhere, or maybe you still don't see the value? All right, great. I like that there's 0 in the last one. That's really great to see. And yes, it's definitely useful everywhere, whether you're in cathode development, anode development, electrolyte, whether you're in novel chemistries, solid state, lithium sulfur, whether you're in non-lithium world of sodium, maybe you're in zinc. There's anywhere that you're trying to count electrons and that those count to mechanistic insights, UHPC is valuable. So we're obviously working on a lot of content around this. So definitely keep in touch with us. Come and check out these continued webinars. The next one, we're going to be talking about quality control, quality assurance. And then in the future, a lot more information about some of our partners. We published some case studies and application notes of some big universities in the United States using our equipment for some of these interesting non-lithium ion use cases. So, check those out. Our website has lots of useful content on it as well that we'll be adding to. So just to reiterate, the applications are everywhere. There's improvements that we're making to our systems all the time. We now have integrated EIS measurements working with Gamry. So all of that's integrated into the software, can be integrated with the hardware. And then, really looking at application relevant testing, not just slow cycling. We have high current models available, protocol integrated temperature control, so you can control your chambers with your protocols. A little more information about the Gamry here. And then even just a little quick case study showing how you can use the Gamry and the UHPC to do measurements while you're cycling cells, while you're getting your coulombic efficiency, your slippage measurements. And you can also be getting really good fundamental insights about your data using techniques like EIS. So I'm going to revisit a slide with just a couple tweaks to it, which is talking about small things with big implications. That's really what UHPC is all about. And just going to reiterate that UHPC can do almost anything a normal cycler can do. And what we recommend is that just a small number of UHPC channels in any battery lab, even if you have thousands of other channels, just a few UHPC channels can make a huge difference when applied to these gaps that other systems have, trying to measure really small things early that kind of snowball into bigger things later on. So I'll pass back to Kevin. Maybe we'll look at the Q&A. And thank you for the majority of participants for sticking with us for a full hour and a half.
Great. Thanks, Stephen. Just to kind of end things out here before we jump to the Q&A, if any of this stuff seems, interesting to you, please let us know. We'd love to get in touch with you. So, you can kind of, select one of these answers here. We can talk about, Voltaiq's offering in terms of an analytics platform. We could talk about Voltaiq's offering in terms of consulting services, kind of what I was talking about in terms of how do you test, analyze, and understand, right? Voltaiq's consulting services help with 1 in 3, the testing and the understanding, right? And Voltaiq's analytics platform helps with number 2. And then obviously NOVONIX has Test House, and they also have UHPC systems, right? So there's lots of different ways that NOVONIX can help out your organization as well. So if you're interested in any of that, please answer this question, reach out to us, right? We'd love to talk to you more about what that would look like for your organization. So with that, there were definitely several questions here. And so I'll ask, most of these are UHPC focused. So I will kind of ask them to Stephen and he can jump in and answer them. So one of the questions that I saw come across was really talking about accuracy. And I know that there's probably different versions of your cycler, but is there any way you can kind of just speak to the accuracy of the NOVONIX systems and what that looks like, milliamps, microamps, those sorts of things?
Yeah, so, this was my first backup slide here in case this question was asked. So, I mean, there's a lot going on this slide. It's all on our website as well. So really when we share a spec sheet, there's a little bit more than those on a traditional spec sheet for UHPC, because we really dive into our source resolution and accuracy, how precisely and accurate we are sourcing current and what the hardware is capable of, and then the actual measurement of that current and voltage that we do. So there's two separate circuits to both source and then accurately measure. So we're not just taking the value that we think we're applying to the cell, we're revalidating it with a precision circuit. And so those two things kind of additively make the resolution and accuracy as high as it is. So, when we're talking about some of these models, so our 2 amp UHPC has a maximum current of 2 amps, and then 5 different current ranges there's 20 milliamps missing here, but 5 different current ranges all the way down to 200 microamps. And each of those current ranges have a full scale, like 20 ppm of full scale source resolution and 0.01% of source accuracy, 20 ppm measurement resolution. So again, all these things kind of, filter down to very, very precise scale. So this 200 microamp is going to have, like tens of nanoamps of precision and accuracy on the 2 amp model in that range. So similarly, the 10 and the 20 amp model, depending on which current range you're in, which will automatically be selected based on what you're applying to the cell, you automatically get these bumps in precision and accuracy. The voltage and timing ranges are here as well. So again, some of these like orders of magnitude better than most of the equipment on the market.
And just to be clear, when I think about ppm, right, am I basically saying like 200 microamps is like a million? Is that kind of how I think about that? Or how do I convert ppm to?
Yeah, it's a good question, because you can basically take it as a percentage, right, and convert it. Yeah, so here it's just we talk about the resolutions in ppm, whereas the accuracies are in a percent.
Got it. Perfect. All right. Another question that came up, you had a slide where you talked about Coulombic efficiency and the definition of that. And then you said it's approximately equal to 1 minus fade minus the slippage, I think was plus the slippage. And there's a question there, and it says, how do you get that fade term? So if you go back a ways there.
A lot of slides.
Yeah, there we go. So 1 minus fade minus slippage. So how do you get the fade term? And I'll kind of add to that question, because I know a little bit that I think could be helpful. Do you do discharge fade or charge fade or charge slippage or discharge slippage? And maybe you can kind of explain and clarify all of that for us.
Yeah. Yeah. And so in here, this is why I've got this little squiggly here, right? It is almost exactly equal to these, and these are terms here. So we've got an application note on our website that we dive a little bit into this. You can also look at the papers by Aaron Smith, Chris Burns from back in 2010 that really split apart all these UHPC metrics and derive them from scratch. And so really this fade term is the fractional fade per hour of the cell, because everything has to kind of be unitless here. And same with the slippage. It's the fractional slippage per hour, where it's, so, the magnitude here of the slippage may be different based on your cell size. A 100 amp hour cell is going to have 100 times the slippage as a 1 amp hour cell of the same chemistry. So there's a bit of normalization in here just due to the capacity and also the amount of time. And so this is, again, approximately equal to, you could take the charge or discharge of each of those: charge fade, charge slippage, or discharge and discharge slippage, because it's really a big combination of each of those terms. So that's why I've kind of generalized it here to these two things. You can think about it here as Coulombic efficiency is equal to, 1 minus the Coulombic inefficiency. And these two things are the Coulombic inefficiency, the two things that are leading to a mismatch between charge and discharge capacity.
All right, great. In one of the slides, I can't remember if it was before or after this, you mentioned that you do UHP cycling, I think it was at 40C, and some of the other stuff was done at 25C. And so there was a question asking, why do you use different temperatures for the UHPC cycling than the standard cycling?
Yeah, and that's a really good question. Typically, we would do the same temperatures if we really want to make sure that we're, especially quantitatively, trying to model something, because it gets a little bit more difficult and maybe you need to use some Arrhenius style approximations if you want to do different temperatures. So in those cases, typically what we do is we'll have maybe a couple temperatures that we're going to test on low resolution cyclers, do lifetime tests, and we'll just do one test on UHPC. And kind of like how we do really nice slow cycling to get nice resolution cycle metrics, kind of kick out kinetics. If we bump up the temperature, we can also test things faster because we see differences in electrochemical stability. We make them a little bit bigger. So this is something that I think Jeff Dahn is really famous for, really pushing the limits of electrochemistry, testing at 60, 70, 80. He's got papers where he's testing cells over 100 degrees to really try to estimate sooner differences in electrochemical stability. So sometimes we'll try to speed that up with UHPC, or sometimes we'll do maybe a study where we'll do a few different temperatures so we understand how slippage really takes off at a certain temperature in a certain chemistry, what coating or dopant, what the limits of that might be in a cathode before we really see a offtake in slippage, if that is a different temperature in different coatings or electrolytes, et cetera. So really, I like to talk about UHPC not as a technique, but a device that you can apply a bunch of techniques to. So it's a good catch that one specifically at a different temperature, but all we were doing is basically making those differences a little bigger. At 25 degrees, they might be a little bit closer together.
Can you just, this is my own question now. Can you track out or extract out activation barriers for different processes, or has that work been tried by doing things at different temperatures and maybe kind of understanding using UHPC, some of these different side reactions and kind of what the activation barriers are and those sorts of things?
Yeah, awesome question. That was actually my PhD. So I was doing a lot of work using isothermal microcalorimetry. So doing really high precision calorimetry measurements on cells with UHPC. So gathering really as accurate of information about the rate of reactions as possible with UHPC, and then separating out the parasitic heat flow off of the cell during those different voltages or temperatures and seeing how the enthalpy of reactions was changing. And in order to do that, you need, as perfect as you can, the rate of reactions, the number of electrons per reaction to do that. And so that was some work that I was doing. We don't have an isothermal microcalorimeter here, or else I may have continued that work. But yeah, that's definitely something that's possible with UHPC, because you can actually accurately say, or as accurately as possible, say, I know the rate of the reactions happening.
Awesome. Okay, so a couple quick questions here. Somebody was talking about when they're going to be deploying 3,000 battery packs, and they were interested in maybe using UHPC to predict lifetime, and just kind of wondered if you had systems that were able to test packs. And I think that you probably answered that with your slide showing the different current ranges and the different voltages. So if your packs fit in that, right, you can test them.
Yeah, and I think it's worth mentioning the same things I talked about with kinetics in terms of, the things that really matter when you can measure voltage and accuracy really tightly is then your system becomes very important. Where your cell testing system is sitting in your room, or even, if you have a pack with 1,000 cells in it, there's a lot of system level noise or uncertainty that might come into play if you're going to test your pack at 1C and try to measure the Coulombic efficiency of the entire pack consistently cycle to cycle. Every cell is maybe at a slightly different state of charge, there's localized heating, which can complicate things when you're measuring voltage to a very, very fine degree. You may actually be at a slightly different SOC cycle to cycle overall in your pack, or have slightly different overpotentials, or your self-heating might be kind of raising your SOC higher or lower based on your measured voltage. So all of those things can kind of layer in noise into the measurement. When you look at your Coulombic efficiency after cycling, you may see a lot more noise than you expect, but it may be because of your system, not because you're not accurately measuring the electrons. You're doing that perfectly, but your system just has the noise built into it. So that's something to mention, which is why, we'll creatively choose whether we're doing slow cycling with UHPC to measure electrochemistry or fast to measure the kinetic effect.
Great point. There's a question about any publicly available open source UHPC data that people can kind of play around with. Does that exist anywhere?
Another great question. Hopefully soon we'll be publishing some of this lifetime prediction work. And the idea there is to also publish the data alongside those methods in paper. But if you want some open source or free to use UHPC data, just send us an email, reach out on our website. We'll be happy to give you some data. I've got, I don't know how many tests are on Voltaiq of our UHPC, but we've got, I think, something like 40,000 or 50,000 UHPC tests. We can spare a couple for anybody.
Great. And then finally, have you guys considered doing anything with EIS data and, doing, high precision EI measurements, or is that kind of outside of your wheelhouse?
Yeah, it's definitely a little outside of our wheelhouse. This is why we've chosen to work with industry experts, work with Gamrate. UHPC is a whole other beast when we're trying to look at, maybe the triangle of test equipment where you can have, precision, there's cost, there's timing resolution, there's these axes that you can't get all of. And so when trying to measure current and voltage very, very, very accurately, but you're also trying to do really rapid things at kilohertz or tens of kilohertz or megahertz, you lose out on some of those other things. So that's why we really try to focus on our testers being high accuracy and precision voltage and current. And then, we're working with experts in those other fields of timing resolution, high frequency measurements, and interpretation of those things. But why we've integrated them is so that people can do those measurements at the same time. You don't have to move your cell from system to system, let it equilibrate in its new temperature, bake it all into one analysis, one system and computer, one channel, reduce your floor space in your lab, and, start using EIS maybe as lifetime prediction features in models.
All right. Final question for our webinar today is, can you distinguish between reversible and irreversible self-discharge? You had a slide, I think, kind of when you were talking about the two differences. Can you actually differentiate between, something that's a reversible self-discharge reaction and something that's an irreversible self-discharge reaction? Can you quantify those?
Yeah, that's a really great question, because there's a few things even looking here, with this manganese dissolution versus a self-discharge or a short in the cell, or this reversible shuttle. Jeff Don did some interesting work recently with PET tape in cells, creating a reversible shuttle which just had this slippage term which just persisted all the way through these cells' life. And that's how they actually identified it, is using UHPC on a cell early in life, thousands of cycles in, and saying, wait, why hasn't this cell died yet? It has all of these additional reactions happening. We're counting all these additional electrons. And, they dove in, did some destructive analyses, which were kind of fueled by the UHPC data to make this really interesting discovery. But when they saw that, they couldn't initially tell, is this just some really mundane electrolyte oxidation that for some reason hasn't killed the cell yet? Or is it all just iron dissolution? I think they were LFP cells. Is it just very slow iron dissolution? Like, back to the envelope calculation, does that make sense? How much iron out of the LFP would that have, come out to be? And so all of that was pointing to, no, it's got to be something reversible. And so that's when, they did those extra tests, GCMS, NMR, and found that this shuttle mechanism was happening. So it isn't easy to tell, because like I said at the beginning, all we're measuring is the number of electrons through this box up here. And what it comes from down here, we need to infer or figure out a different way, maybe through an analytical method or a theoretical method. So there's that extra layer of insight that we get here, but then there's still a limitation without opening up the cell or doing more destructive or, looking at the cell maybe with a beam line or something, to see what's going on. So I wish I could tell you, yes, this magical technique will give you whether it's slippage or a reversible shuttle. Again, in some of the work I did in my PhD with calorimetry, we did some shuttle work to show net zero enthalpy reactions. But again, that was another method, another technique and measurement that was done to complement. So UHPC may not give you all of the information and say your EC is breaking down, but it will tell you if something's happening and it's happening here or very likely here and at this rate.
Yeah. And that's part of the reason why, it's a whole host of tools and techniques, right? Everything is I like to call it a tool in your toolbox, right? Like UHPC is a great tool in your toolbox, right? It helps you solve some of these questions, right? Then rest step analysis, another one. Like maybe rest step analysis, you could look at the transport and you might be able to see differences between an irreversible reaction and a reversible reaction and how it affects, motion across the cell in terms of concentration, relaxation, and things of that nature, right? So there's different dynamics at play that when you put them all together, right, help you get to this mechanistic insight that we all desperately need to help us make, great informed insight from our data, right?
Yeah. And, I'll be the first to say that my lab here has 3,000 test channels in it, and only about 10% of them are NOVONIX UHPC channels, because we're choosing what we do with those channels and putting them to use where they are most valuable. And the rest of the channels in my lab are, just all these other types of brands. We have, I think, 5 or 6 different makes and models of cyclers here. Some of them, I think, are even folks from those companies are even on this webinar watching, right? So we understand the need for all these additional tools, and that, there is such thing as a balanced lab. We don't think anyone should have an entire lab full of 1,000 UHPC channels. It would be wonderful, but it's really not necessary. Choosing where the most value add for that application is, and really just making the use of those, the best use of those channels is what we really strive for.
And Voltaiq can help you organize it all. Well, thank you so much for joining us for the webinar today. We really appreciate it. We'll be back next month with our final part 3 of this series. So with that, thank you all for joining and have a great rest of your day. Bye.